Okay. So we looked at this slide already. These are the 7 functional groups that you want to be familiar with. And remember, a functional group is just a group of atoms that you see grouped together a lot, stuck to a molecule, stuck to a biological molecule. So these are common combinations of atoms that you'll see put together. I think I got through carboxyl. So I was going through each of these kind of individual images. So we got hydroxyl, carbonyl, and carboxyl. This is where I made a note to myself that we ended. So we looked at carboxyl, that double bond to the O, C double bond to O is the giveaway for, and then an OH, sorry, is the giveaway for carboxyl. So you have an OH. You can ignore this right here. Don't worry about that ionized form. Don't worry about that part on the right. This is what I would ask you. I'd give you something that looks like that and say, what functional group is this? So the C double bond O with an OH coming off of the other side of the carbon. And remember, the stuff at the bottom, don't worry about. It's just additional explanation in all of this text. I really want you to know the name and be able to identify an image of the functional group. Okay, so we're going to continue on with the rest of those functional groups and then get into a little information about carbohydrates today. All right, next we have amino. And I mentioned amino is easy because this is the only one that has nitrogen in it. So N plus 2 H's coming off of either side of the N makes it an amino group. Obviously important in amino acids if you've heard of those. You'll become very familiar with amino acids when we get into proteins. They make up proteins. And again, don't worry about the ionized form. That's a little beyond what you guys need to be familiar with. So for you guys on Zoom, don't worry about this part over here, the ionized form. All right, so NH2, that's amino. And then sulfhydryl. It's the only one with sulfur in it, I think, so that's easy. Sulfhydryl is just an S and an H. You can see it right here, SH. We don't come across that one all that much. It is in some amino acids, but this isn't all that common. Phosphate. Phosphate we'll talk about a lot throughout the semester. So familiarize yourself with the phosphate group. It's basically phosphorus with 4 oxygens around it. This is kind of a weird way to write it, but it's phosphate with 4 oxygens, one sort of on each side of the phosphorus. So here you can see, and you don't have to worry about the double bond necessarily, just know it's phosphorus. You see phosphorus, it's a phosphate group, basically, with the 4 oxygens around it. Okay. So why we care about phosphate is because, well, it's in a lot of different molecules, but ATP has 3 phosphate groups on it. And you're not familiar with ATP yet, but you will be once we get into some of the cellular functions. This is the main energy source for our cells. ATP is like the fuel for our cells. So you'll see ATP a lot. It stands for adenosine triphosphate. So that means 3 phosphates. And those 3 phosphate groups are basically what gives it all of that energy. So that's a huge reason why you will continue to hear about phosphate throughout the semester, because we'll be talking about ATP a lot. And then finally, we have the methyl group. This is just CH3, so 3 H's bonded to a carbon. If you're familiar at all with methane, methane is CH4. It's a molecule, a greenhouse gas, basically. So methyl is just missing one of those H's, CH3. You don't have to know methane, but I figured if for some reason you knew methane, it makes it a little easier to remember the methyl group. Okay, so those are the 7, I believe, functional groups that you need to be familiar with. Just be able to identify them. If I gave you an image of one, or I told you what atoms or elements were involved, be able to give me the name, or vice versa. If I give you the name, tell me what atoms or elements are involved in that functional group. All right, any questions about those? What you need to know with them, or any details on those functional groups? All right, we'll see them in some of these molecules, these big molecules we're about to talk about. So that's why I brought them up. So now we're going to get into what we call macromolecules. Macro means big. So micro is small, macro is big. So these molecules are big molecules. And they're essentially, as I said here, the building blocks for life. So what are they? You've probably heard of all of these, at least maybe sort of just in passing, or in a more casual rather than scientific area. So we have 4 categories of these macromolecules. I'm going to go through each one and talk about their characteristics and why they're important from a biological perspective. So we have carbohydrates. I think everyone knows that word. So we'll talk about what carbohydrates really are. We'll talk about lipids, which are basically fats. So oil and butter and all that good stuff. Those are lipids. Proteins. Everyone's pretty familiar with proteins. And then nucleic acids. Those are probably ones that you're less familiar with, but that's our DNA. DNA is a nucleic acid. So I do want to talk about some characteristics or some features, I guess, that all 4 of these sort of have in common. And we'll talk about how those features differ between each category as we go along. Yeah, so all 4 share some common features. We'll talk about those common features and then move into talking about each of these types of macromolecules specifically. Common structural features. Number 1, they have a ton of carbon. So these are organic molecules, and organic pretty much just means there's a lot of carbon. It's carbon-based. So lots and lots of carbon. They also have hydrogen and oxygen, and for the most part, those are the only atoms that are involved. With amino acids, obviously we'll have the amino groups, so like NH2. There might be some sulfur in there, some phosphate, but for the most part, the vast majority, like the big chunk of these molecules, are all carbon, hydrogen, and oxygen atoms. So C, H, and O. And their structure. So in general, their structure consists of all of these little individual units that are bonded together. So we can break them down into what we call monomers. And we'll talk about the monomers. Mono means one, poly means many. So a bunch of monomers strung together makes a polymer. And they're bonded with covalent bonds. So they're sharing their electrons to make these really long strings of these repeating units. So kind of the same thing over and over. Smaller pieces get strung together to make the polymer. And we'll get into detail and I'll give you examples of what that means. But just think of it as sort of individual pieces. I think I might have an image here, yeah. Like individual little pieces, this would be the monomer. You string a bunch of them together with covalent bonds and you get the polymer, which is going to be, this is going to be our macromolecule, the polymer. A big, long, strung together polymer is our macromolecule eventually. And these bonds are basically made and broken in the same way, through the same reactions, whether it's a carbohydrate, a lipid, a protein, or a nucleic acid. So no matter what category the molecules, the polymers fall into, the monomers will be bonded together through a specific type of reaction that I'll talk about, and they'll be broken apart by this, a specific reaction as well. So across all four of those categories. Okay, so how do we do that? How do we make polymers or break them down? There's two reactions. So one to make them, one to make the polymer, and one to break the polymer. So in order to string multiple monomers together to make that long chain, that polymer, it's a reaction called a dehydration synthesis. So synthesis means to put together. Dehydration means water is basically being pulled out. And I'll show you sort of what that looks like in a simple sense on the next slide. So at the end, you have a polymer, which is really important because that's the whole goal, is to synthesize a polymer. But then a byproduct is water. Like I said, water is pulled out. That's why it's dehydration. It's dehydrating the individual monomers, I guess, is a way to think of it. Pulling water out, and then the polymer is the goal, what we're trying to get to. If you want to, you have a long polymer, like a long string put together, and you want to break it, it's a reaction called hydrolysis. So we're still dealing with water, but now we're using water to split those bonds. So we're basically putting water back in to break those monomers apart. Hydro obviously stands for water. Lysis means to split. So anytime you see lyse or lysis, that means to split something. So here we're moving from a polymer, breaking it down back into its individual monomers, its subunits. And interestingly, so the product here would be the monomers. The reactants are basically the same as the products are in the dehydration synthesis. So here the reactants, what's going into this is the water and the polymer. That water is going to break that polymer apart. So you just have these individual monomers at the end. So it's interesting that the products and the reactants are the same, but on opposite sides of the reaction for each of these types of reactions. So definitely know what the products of dehydration synthesis and hydrolysis are, as well as the reactants. So if you look up here, the reactants would just be the monomers, right? So you're taking the monomers, putting them together, that results in water and a polymer. Here we have water plus a polymer, and you end up with the monomers. So hopefully this next diagram helps sort of explain it. I'm going to break it down, so don't get overwhelmed by the text and everything. So in the upper left here, we have this short polymer that's being built, right? But we want to add another monomer to it. So we want to attach this monomer to that growing polymer. And basically at the end of these individual subunits, these monomers, there's an H and an OH. And there was an H and an OH between 2 and 3 here, and there was one between 1 and 2, but they get taken out, basically, and that's H2O, 2 H's and an O, right? So you pull out those atoms, that makes water, and then it allows this final unit to bond on to the growing polymer. So you can imagine it as if you're like squeezing water out of those two individual pieces and allowing it to bond with water being a byproduct. So for you guys on Zoom here, this H and OH, that's going to make water. So pull that out. It's just a byproduct, and then that allows that 3 subunit to bond to this new unlinked one. So you have the 3 to the 4 down here. Does that make sense? Okay, you're pulling water out. That means you can then stick those 2 individual pieces together. Okay. If we want to go the other way, hydrolysis, we want to break down a polymer. So we have this long string, but we want to cut one off. You add water in. So that's what this is showing. So we have water, it's being added in, and then that's going to break that bond, basically. So water will kind of dissolve that bond. I guess that's a way to think of it. So then you're back to what we started with up here. I know there's a lot going on this slide, but it actually does a pretty good job of sort of simplifying and explaining exactly what's going on. Dehydration removes water, forming a new bond. Hydrolysis is adding water, which is going to break the bond. Really straightforward and as succinct as possible in a somewhat complicated reaction. So no matter what kind of molecule you have, a carbohydrate, a lipid, etc., they all go through the same process to be made or broken, these same 2 processes. Okay, so now we have some of the background on what these macromolecules are. And now we're going to jump into carbohydrates, talk about their structure as well as their function. Everyone's heard of a carbohydrate, right? We know that sometimes carbs are good to eat and sometimes they're not good, and there's all this debate out there about whether we should eat carbohydrates or not. We do need carbohydrates. All animals need carbohydrates because it's one of the 4 major building blocks of our body. I guess if you eat too much of them, maybe it's not good, but we're mostly just going to be talking about it from a very simple structural standpoint. So carbohydrates, if you break them down, so they're typically these really long, complicated molecules. Their individual subunits are called monosaccharides. That's their monomer. So you put a bunch of monosaccharides together and you get this long carbohydrate. And the chemical formula, so in carbohydrates, there's only CH and O. There's only carbon, hydrogen, and oxygen. There's no other atoms, only CH and O. And there are always some multiple of this formula. So for every 1 carbon and oxygen, there's 2 hydrogens. So you double the number of hydrogens compared to the number of carbons and the number of oxygens. Glucose is one of our main carbohydrates in our body and one of the most common carbohydrates on Earth. It is a monosaccharide, so it's just one little subunit. And its molecular formula is C6H12O6. So it has 6 carbons, 12 hydrogens, and 6 oxygens. And I'll show you what that looks like in just a second. But you do want to memorize that formula, C6H12O6. That's glucose. You'll see it a lot. So in addition to glucose, there's a few other monosaccharides that you need to know. So these monosaccharides, remember, these are the monomers. They're just simple subunits. So there's galactose, fructose, and ribose. And when I say glucose is C6H12O6, this is what that would look like if you drew it out. So you can see all 6 carbons, they kind of form that backbone. Carbon is usually going to be the backbone of all of these organic molecules. You have some H's and OH's coming off. And then you have this C double bond to O up here. So that's our carbonyl group. So that's one of the functional groups that you should be familiar with. And you're probably thinking, all of these look really similar, right? So I have all 4 of these molecules that I listed over here. I have the diagrams of them over here. So I want you to be able to identify, based on these types of diagrams, what monosaccharide you're looking at. So I'm going to start with ribose. Ribose is easy because there's only 5 carbons. So 5 carbon sugars, there's only 5 in the middle. So if you only have 5 carbons, that's ribose. If you have 6 carbons, it's one of these 3. So you can break it down that way, at least start out, eliminating 1. Ribose is very easy to identify. All of the OHs are coming off of one side, the H's are coming off the other side. It's very uniform. You still have that carbonyl up top. So we have a carbonyl group in each of these. So ribose, 5 carbons. When we get down to here to the 6 carbon ones, fructose is the other one that kind of stands out, right? So the carbonyl group here in fructose is not off of the end. So it's on the 2nd carbon down, carbon number 2. For the other 2, that carbonyl group is coming off the very end. And I know that seems like a really minor difference, but it changes the characteristics of this molecule just by changing where that one bond occurs. It's kind of crazy. And then, so the tough ones to distinguish are glucose and galactose. So still 6 carbons, carbonyl group coming off the top. The only difference here, crazy enough, is that the hydroxyl and the hydrogen are on opposite sides. So glucose versus galactose, you just flip the H and the OH on the 1, 2, 3, 4th carbon down. And I probably won't ask you to distinguish between these 2, but I will likely ask you to distinguish between one of these and one of these 2, or these 2 between each other, that kind of thing. I think glucose and galactose are similar enough that I'm not going to ask you guys which is which. Does that make sense? So just know that like glucose and galactose, you got that carbonyl coming off the end and it's 6 carbons. There's 6 carbons in here. Okay. In lab, I think this week, maybe, you guys are going to be doing biological macromolecules. So you'll actually look at them more in ring form. So you don't have to know these rings for lecture, but do know, and I just want you to be prepared for lab. These straight line structures, that's not how they occur in living organisms. They typically bend around and form a ring. And you guys will make that in lab this week, I think. So you'll deal with the ring structure more so. That's sort of how they are found in living organisms like that. But you don't have to know those for lecture. I just wanted to point that out and make you aware of that. This is what we'll be focusing on because I think it's a little bit easier to see and understand compared to that. So for an intro bio class, I think that's fine. Any questions about this? Okay. Now this is the one type of macromolecule category where I'm actually going to talk about just 2 monomers stuck together. Usually we talk about the monomer and then the really big polymer. With carbohydrates, there's a few important disaccharides, which means two monomers put together, so just two. So for carbohydrates, we have the monosaccharides, disaccharides, and then the polysaccharides will be the really long chains. So it's a little bit unique for the carbohydrates here. The bond between any two monomers, so this goes for disaccharides as well as when we get into like the really long polysaccharides, the bond between two monosaccharides is called a glycosidic linkage. You're going to want to know that term. Glycosidic, kind of like glucose, but slightly different, but it sounds sort of like, glucose, so hopefully that helps you remember it. Glycosidic linkage is what's going to bond two monosaccharides together and is done through that reaction I talked about, dehydration synthesis. Every time we bond two monomers together, it's done through dehydration synthesis. That's the reaction type. The bond name will change based on the category of molecule we're talking about, but for carbohydrates, it's always a glycosidic linkage. So a couple of important disaccharides, maltose and sucrose. So you see a lot of the oses on the end, glucose, galactose, it's all ose, right, with the carbohydrates. That ose just means sweet or sugar. So all of these sort of have like a sweet flavor to them. Maltose is when you have two glucoses stuck together. That's used in making grain products, including beer. So like a malty beer, if you've ever heard that term before, it's because of the maltose in there, it's giving it that flavor. And then sucrose is table sugar. So when you put sugar on or in anything, you're using sucrose, which is actually a disaccharide of glucose plus fructose. Here's an image of maltose. This is kind of what two rings look like when they're bonded together. Just for your information, you don't have to be able to identify that. Just know the names of these two disaccharides, what they're used for, and then the two individual units that make them up. So glucose and glucose is maltose, glucose and fructose is sucrose. A lot of oses. All right, then we get into the polysaccharides. So poly means many. So now we're putting a bunch of those monosaccharides together. So more than just two, basically. Bond, same name, glycosidic linkage, same as with the disaccharides. That link between each monomer subunit is still just a glycosidic linkage formed by the same reaction, dehydration synthesis. So nothing different, just many more pieces put together now. And there are 4 important polysaccharides that you should be familiar with. I'm not going to talk about their structure, like what individual units they're made of. I really just want to talk about their function, and then, well, I have a little bit about some general structure stuff for, I think, cellulose and chitin. So starch, you've probably heard of starch. It stores energy in plants. So if you eat a potato, that's a lot of starch, and it's basically all of this energy that's stored by that potato plant as carbohydrates. So that's starch. Glycogen is how we and other animals store carbohydrates. So we don't have a potato in us, but we have glycogen, which is how we take the carbohydrates and store them for energy to be used later. The last 2 are structural, and by structural, I just mean they provide more solid structure to the organisms that they're in. So cellulose is in plants. It's structural. It helps plants kind of stay it's part of the reason why plants stay upright and their cells are pretty tough. And then chitin, which you may or may not have heard of, is structural for fungi, so mushrooms, as well as a lot of invertebrate animals, so insects, lots of crustaceans. All of their exoskeleton, the outside of them, that hard part, is chitin. So 2 storage types of carbohydrates, 2 structural types, 1 for plants and animals, essentially, in each category. So I want to talk a little bit about cellulose and chitin, just in a little bit more detail, those 2 structural ones. Cellulose is particularly strong due to hydrogen bonding between polymer chains. So what that means is you have all of these, see these individual subunits put together. So these are the rings, we'll say it's all glucose. So if it's all glucose strung together, these are the glycosidic linkages, right? They're bonded there. They also have hydrogen bonds. If you remember back to hydrogen bonds when I was talking about them, they'll actually bond between these individual chains of glucose. So that kind of creates this lattice of really strong cellulose. So when you're eating dietary fiber, like celery has a lot of dietary fiber, it's really hard for us to break down and really hard for a lot of animals to break down. So it actually, that's why fiber is good for you. It basically doesn't break down and cleans out your digestive system. So that's all due to the fact that there's these hydrogen bonds bonding these individual polymer chains together into this really complicated, not complicated, but highly structured, strong cellulose molecule. All right, so that's cellulose. That's all you really need to know about cellulose. And then chitin, since I study invertebrates, I think chitin is particularly important. It's a really strong polysaccharide. It makes, like I said, the outer shells or the exoskeletons, the outer skeleton of invertebrates. So these lobsters here, they have a lot of chitin in their shells. And I found this really interesting article, I think last fall, about students actually taking those lobster shells because they're really tough and it making them into biodegradable packaging. So chitin, totally natural and biodegradable. So rather than using, say, plastic, I don't know how they did it because I didn't actually read most of the article. I just found it. But they took that idea, the chitin idea, used some of the exoskeletons and started making biodegradable packaging. And I should look into how far that's gotten, if it's actually gotten any traction, but I thought that was particularly interesting. All right. Let's just barely start lipids. We're just going to get into lipids. And then on Wednesday, I know this is a lot of talking today. I'll kind of break it up a little bit on Wednesday and have you guys sort of summarize some of this information with a partner in class. But rather than trying to squeeze that in right now, I think we'll just start getting into lipids, do that on Wednesday. Okay, so lipids. Lipids are sort of, they're not as nicely organized as carbohydrates where you have monosaccharides and disaccharides and it's all like clear cut. Lipids are sort of just loosely grouped based on the fact that they don't mix with water. So I'll talk about, I think, 4 different categories of lipids. They all are significantly different from each other, but none of them mix with water. So you can think of oil. When you think of a lipid, think of like olive oil. They're hydrophobic. That means water fearing. They are scared of water. Now, there's not really any distinct monomers that you can pick out. They're just really long chains of mostly hydrogen and carbon. So yeah, 4 main groups that I'll go through fairly quickly. We've got fatty acids, triglycerides, phospholipids, and steroids, or sterols. Fatty acids and triglycerides are actually pretty similar. Phospholipids are important in cell membranes. We'll get to that. And then steroids and sterols are a lot of hormones and cholesterol. Cholesterol falls into the sterol category. Okay, so we're gonna first talk about fatty acids and triglycerides. Oh, but first, an important point is that these have a lot more energy per gram than carbohydrates. So they can store a lot of energy in an animal body, or plant, I guess. So lots of energy storage potential compared to carbohydrates. All right, so what are fatty acids? Fatty acids they have a carboxyl group on one end and then a methyl group on the other. In between, it's just a whole lot of carbons and hydrogens. So here we have carboxyl. That's one of the functional groups you should be familiar with. So C double bonded to O, and on the other side, you have that OH. On the other side, you can see the methyl group. So you have C and then 3 H's. So that's going to be methyl. So if you see a long molecule of a bunch of carbons and hydrogens with those 2 caps on the end, you know it's a fatty acid. And we can talk really quickly about saturated versus unsaturated fatty acids. So you guys have heard of saturated and unsaturated fats, right? Do you know which one is supposed to be less good for you, like less healthy? Saturated. Saturated, yep. So saturated fats are not as good for you because basically, I'll get into this. I'll talk about this again because I'm going to go through it sort of quickly here since we're almost at the end of class. The saturated fats are solid at room temperature, which means they might be solid in your body as well. We don't want solid fats in our arteries, right? So saturated fats, solid at room temperature. Unsaturated fats are liquid at room temperature. And they have a slightly different structure. And I'll start with that on Wednesday, why this makes them liquid at room temperature and this makes them solid at room temperature. Just one tiny, tiny difference. So we'll start with that on Wednesday.